Hi class, it is Mr. McGregor.
And Mr. S. Okay, so what you're looking at here is the famous Griffith experiment. And this is actually the experiment that showed that DNA is actually a molecule used by the body to give direction, especially to the cells. Now, in this experiment, what had happened is you had two cells. You had the S cells and the R cells. When you injected the S cells into a mouse, the mouse died from a infection. When you injected the R cells into the mouse was able to handle it. Not a big deal. What they did is they then killed the S, or the killer bacteria, into a healthy mouse, and because they had heated up the S killer cells, they killed it. And so since the bacteria was dead, when they injected it into the mouse, nothing happened. It didn't kill the mouse. The interesting thing happened, though, when they mixed the R cells, which were the cells that didn't kill the mouse, and the dead bodies of the S cells. When they mixed those two bacteria, they had found out that the mouse were then dead. So the hypothesis, and what eventually got proven true, was that the directions from the S cell, the killer cells, so the DNA from the S cells actually had gone into the non-lethal R cells. And when that happened, it killed the mouse.
So to help make a little more sense of that mouse experiment and some of the genetic content that we had just gone over in the last unit, we're going to describe and talk about the basics of DNA and a little bit of its history. DNA is a molecule that is responsible for everything that genes do. The genes in your body are the things that code for proteins and determine what traits you have. And genes are little segments of DNA. So DNA, as you know it, and I'm sure you guys have seen, we'll talk more about the structure of it, but DNA kind of forms this structure that you guys are all familiar with called a double helix, and the genes are just little segments that make up that structure. So DNA needs to do a few things. It needs to carry information, genetic information, from ancestor to ancestor. It needs to be able to make that information become a reality, so give physical properties based on that information. And it needs to be easily copied over and over again. And so the analogy we like to use is when we think about computer files and how we transfer them. Sometimes we'll use a flash drive to take information from one computer, copy it, and move it to another computer without losing any of that original information on either A, the first computer, or B, the hard drive. So that's a good way to think about it. And if we want to bounce that back to the mouse lab for a minute, when both the dead lethal bacteria, the S cells, and the non-lethal bacteria, the R cells, were combined into one mixture to be injected into the mouse, those non-lethal bacteria actually had the DNA information from the lethal S cell bacteria encoded inside of it. So it basically took a non-lethal bacteria and copied that genetic information from the lethal bacteria and became lethal itself.
So DNA, it's a very special molecule, and just starting with the basic structure of DNA. Now, DNA is composed of many of these, but if you remember back to the macromolecule unit, nucleic acids are composed of a monomer called nucleotides. So this is a nucleotide. So this is just one of, this is one building block that when stacked up becomes DNA. But just looking at the single building block, the monomer, a nucleotide is composed of three main parts. You have a phosphate group, which is that circle, a deoxyribose sugar, that's where the D in DNA comes from, and then it has a nitrogenous base, that rectangle.
That shape that Mr. Rest just talked about, the nucleotide, can have 4 different possible nitrogen bases. So we section them in different groups called purines and pyrimidines, but the 4 different possibilities are adenine, guanine, cytosine, and thymine. Now, adenine and guanine are called purines. They have 2 rings, so you can see right here. And pyrimidines have 1. One of the mnemonic devices to use to think about this is a pyrimidine starts out kind of similar. It sounds similar to pyramid, and a pyramid is a single geometric shape. So if you can kind of equate pyramid and one geometric shape with the single ringed nitrogenous base, either cytosine or thymine, that's a good way to remember it. From here on out, we're really going to refer to these by their letters. So we'll be talking a lot about A and G and C and T. We might not use the full terms that frequently. So as long as you remember A, G, and C and T. Also, I'm going to point out here that we have 4 different nucleotides happening. So we have a nitrogenous base here, 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 and here. We stack these up to kind of show you what they look like when they're connected. Here is another picture of them not connected. So you'll fill in for adenine, you'll put an A in this nitrogenous base right here, a T for thymine, a C for cytosine, and a G for guanine. These are the same as the ones in the previous slide, but they're just not connected to give you a bigger picture of what they would look like. We also don't show which are purines and which are pyrimidines in these. Just keep that in mind. Also note the fact that each one of these nucleotides has a phosphate group and a 5-carbon sugar.
Okay, and so back to that image from before of the 4 nucleotides stacked on top of each other. The thing that actually keeps the nucleotides together, like with Legos, you know, they got those little hump things on top of each other and they actually connect that way. Well, how do these nucleotides stay together? It's right there. It's that little stick. That stick is supposed to be a bond. Here's another one right there. And those are called phosphate bonds, okay? And what we call it is a sugar phosphate backbone in the DNA. It's called a sugar phosphate backbone because you'll notice that this phosphate attaches to this sugar right there. And so that's called the sugar phosphate bond. And that is what connects all nucleotides together. So the backbone, the thing that kind of holds the DNA together, just kind of like your backbone holds your limbs to one structure. So the backbone of a DNA molecule is that sugar phosphate group. So going back to before DNA was actually understood to be the molecule that transfers information not only in your cells, telling your cells what to do, and they give you your traits. DNA is also the thing that's inherited from your parents and all that. But before that time, DNA was known. They knew about deoxyribonucleic acids, but they did not understand what their purpose was in the body. We're going to actually do a lab with this, and it looks like snot or something. DNA is kind of gross looking. And there was this guy named Chargaff that liked to experiment with that DNA-looking snot. And Chargaff had noticed that when he analyzed DNA, each nucleotide, if he found a nucleotide that had a nitrogenous base of guanine, there would always be a match for that nucleotide with a nitrogenous base of cytosine. And the same thing with adenine and thymine. So essentially what happened is you could imagine that, if there were 50 guanines in a amount of DNA, without even measuring the amount of cytosine, he could guess that there would be 50 cytosines. And then upon analyzing the DNA snot glob, he would see that he was right. And so this became a very interesting thing, that the number of adenines would always equal the amount of thymines, and the amount of guanines would always equal the amount of cytosines. But no one knew why this was. It's essentially, imagine yourself kind of walking into a high school dance, and you notice that there are 25 boys, and then there are 25 girls, and you didn't know why, but there's I counted 25 guys and 25 girls, and I have no idea why. That's pretty much what Chargaff did. It wasn't until later that we found out why the numbers of guanines and cytosines would equal, and the amount of adenines and thymines would equal.
Another key figure on the road to the discovery of DNA and its structure was the British scientist Rosalind Franklin. So she did most of her work in the 50s, the early 50s, and she looked at DNA by using this technique called X-ray diffraction, which is a fancy way of talking about taking these sort of like photographs using X-rays to kind of capture the shadows cast by really tiny, tiny molecules. So her pictures showed that DNA had this spiraling X structure called a double helix. Here it just says helix, but I'm going to write in double. All right. And to just kind of unwrap what a helix is, a helix looks like the coil in a spring. So this looks different than the structure you know DNA to be, because a double helix is like two springs going in opposite directions, and it forms this kind of like spiral staircase looking structure. Now, unfortunately, even though at this point in history we're 30 years into and past essentially the significant women's rights movement, being a female scientist in a particularly male-dominated field was really challenging. And so Rosalind Franklin at the time was not credited for her work in helping to find the structure of DNA. And at the time, there were kind of two camps, both vying, fighting to kind of figure out the mystery behind the structure of DNA. And on one side, you had Rosalind Franklin, and on the other side, you had these two guys called Watson and Crick. And Watson and Crick had all these kind of little pieces of the puzzle, and they were trying to put it together to figure out what the structure looked like. But really, without her X-ray diffraction photographs, they would have never been able to figure it out, and they never gave her any credit for her work.
So Watson and Crick, the guys that were just mentioned, who had actually used Rosalind's photo without her knowledge, actually. So that adds another level to it. Not only was she not credited, she actually had no idea that they had stolen her image to figure out the shape of the double helix, and they actually used that to build a model of the double helix, and it is the model that we know and it has been proven correct today. You can actually see the double helix structure here, where Ms. McGregor just had mentioned that a helix is just like one spring spiraling. You can actually see here the double helix, both interweaving between one another. And we definitely say it looks like a spiral staircase because each of the nitrogenous bases make up kind of like steps, which are shown right here. And so this is called the double helix because it's two helices interweaving between each other. So Watson and Crick not only used her information, but like most scientists, he also built upon the back of Chargaff's. If you remember Chargaff, he was the one that saw that the A's and T's were always an equal number and the G's and C's were always an equal number. It was Watson and Crick that actually put it together that the reason the A's and T's and the C's and G's always equal in number is because they are the rungs on the ladder. They need to come together in that perfect ratio, that 1-to-1 ratio, to be able to make the rung complete. As you can see over here, the rung that is seen in this image is in this image shown. You can see the C's and G's are connecting to each other, and that's making one of the rungs. The G and C here connecting to each other, that's another rung. AT, that's another rung. TA, that's another rung. You can actually see the sides of the ladder are the sugar phosphate backbones. So there's actually two sugar phosphate backbones in the DNA molecule. That's shown right here. And if we were truthfully going to say, we would simply, if we were going to simply say it looks like a ladder, but then if you take that ladder and you twist it, now all of a sudden you see it's the double helix. Something important that you must know is that the bases are held together by a special type of bond called hydrogen bonding. You might remember this from the water unit. It's the same type of hydrogen bonding. It actually happens inside the nitrogenous base itself. Remember, the nitrogen base doesn't actually look like a shoebox type rectangle. It's actually atoms that are inside there. The atom's hydrogen in the cytosine are connecting with the hydrogen atoms in the guanine. And there's actually three connecting with each other, and that's why there's three dotted lines. Between adenine and thymine, the A and T, there's actually two hydrogens connecting with one another. Two sets of hydrogens connecting with one another, and that's why there's two dotted lines right there. And finally, finishing up my analogy with Chargaff. Remember, Chargaff kind of walked into a high school dance and he saw, well, in this high school dance, this particular one, he saw that there was 25 boys and 25 girls, and he didn't know why. Watson and Crick were the people that said, well, at this particular dance, it turns out that the 25 guys are pairing with the 25 girls, and that's why they are an equal number, because they actually pair up with one another.
Tomorrow's Bell quiz will be these questions. So, what does this ladder represent? What are the rungs made of? What are the sides of the ladder made of? And finally, what would need to happen to the ladder to make it look more like DNA? So you guys need to draw these in your notes. Just a little quick pointer on drawing a double helix. Start off with just one helix, and then draw the other one to match, but just like, an inch away from the other. All right. So kind of in the pattern that I just did. Now it looks terrible, but yours will look better than that. And then just draw the rungs across. And then this other one, I think you can figure out, but make sure you have the hydrogen bonds in there because the number of them is important. I was going to wait and give you guys a bunch of time, but actually just pause the video and draw it, and then turn the video back on.
Okay, now that you've drawn the DNA, you are looking at right now just a stack of nucleotides. That's supposed to be represented by these Legos. So you have a stack of nucleotides. Okay, that would just be supposed to be just a single strand of DNA. But remember, the DNA is a double helix. So go to the next slide and we've actually added a second column of Legos to make it the double helix, and I want you to match the nucleotides together. Okay, trying to remember which of the nucleotides go together and match it up.
So that's it for today's notes. We will see you in the next video. This is Mr. McGregor.
Mr. S.
See you guys in class tomorrow. I mean, we're in class right now while you guys are doing this. But we'll see you also tomorrow.